audiences to come in the front seats so you can hear better and the sound reaches you with a better quality. I have another announcement to make. The last session, the very last session of today, will be run in parallel. And the idea is to finish the program a little bit earlier. So the last session, only the last session, will be run in this hall and in one of the smaller auditoriums. I would also like to inform everyone, including the ladies up there, that you are most welcome to come and see the posters. There are more than 100 posters, very high quality posters. So please stick around, and the presenters are always keen to answer any questions or show their work that they've done. So please take the privilege and freedom to come down, visit the exhibition, and visit the posters during the break time. Right. We'll start with our first session, and I would like to call upon the chairing persons. Professor Sudhir Kumar, please. And I will be with him. So hope you enjoy.
Welcome to the morning session. And since we are already running a few minutes late, let us get started. Today we have the first talk from Professor Emmanuel Dermatitis, and he will discuss, he's a professor of genetics in Geneva Medical School, and he will discuss his work on, this should change, it's about phenotypic evolution, actually the effect of genetic variation on phenotype in the hereditary regions. So, Dr. Manolis is coming.
Okay, well, thank you very much for the invitation and the opportunity to be here. It's a great pleasure, and I hope that some of the work that I present today is going to be of interest to people here, to see some of the capabilities we have in actually using omics technologies to get into understanding biology, basic processes, but also try to see how this could have an impact potentially on understanding health-related genetic effects and how we can use them forward. Now, one of the fundamental issues of understanding complex traits, and with the realization that pretty much every trait that you have in a human population is complex, is that we have a number of different levels at which one can measure phenotypic effects. So, on the one hand, you can imagine that you have, of course, you have the DNA variation, which is inherited at -9 months of somebody's life. And then at a higher level, you may have, the cell effects, whatever happens in the sort of things. Very useful. You can see that, I hope. Okay, so you have DNA variation inherited at very early on, so it's actually much earlier than any environmental exposure that this organism has. But you also have the effect that is represented in the cell, which actually is, at some point, the joint effect of the genome plus whatever environment that individual has been exposed to, and whatever epigenetic and other permanent or transient effects are actually operating in the cell. And all these things, of course, summarize into an organ tissue level, and at the end, over the whole organ space and tissue space of an organism, plus the time space, the time dimension, so the temporal aspect of all these effects, is leading to a particular phenotype, which we can classify as disease. Now, as you realize from this, there's two very long and complicated processes. That's the complexity of the interactions with the cells and tissues, but also the accumulation of the signal from the environment. And how this jointly contribute to the disease cannot be simply described by the proportions of variance explained by one or the other. It has also has to be taken into account in the context of how they interact and how one talks to the other one. And in many cases, it is expected that the impact of, for example, an environmental effect has to do a lot on the underlying genotype of an individual. So it's not a sort of just adding effects, it's more trying to integrate them into a single model and try to understand how they jointly have an impact in any particular complex trait, regardless of whether it's a disease or not. Now, one way to think about integration is to think about networks, and people have been, you have seen all these hairballs published in big journals, where you basically get a degree of correlation of measurements that one can make. So this correlation can come either from measuring, for example, gene expression or quantifying protein-protein interactions. And by doing that, you can actually develop some level of relationship among specific types of genes or other types of measurements. Now, one of the problems with all these networks is that you have this correlation structure, but very little information about causality. And in order to go into causality, essentially putting an arrow to a direction of an interaction, it needs very laborious experiments that cannot actually cover the whole space that we're exploring, let's say all genes against all genes and so on. Now, an alternative way to think about networks is to think about phenotypic networks. And in many ways, the co-expression networks that we've been thinking are phenotypic networks. So we can think of phenotypic networks as a way to derive a correlation structure among phenotypes that we can get from the same a set of individuals. So what you get, for example, is how a certain metabolite is correlated with blood lipids and so on. Now again, this has a correlation structure. It doesn't give you information about causality, but there's one fundamental piece of information that can actually give you, at least in a statistical way, a direction of that effect, and that is DNA variation. Now, for the most part, actually I would say almost in all cases, except potentially for cancer, it's very clear, and it's actually fundamental, it's sort of an axiom in biology that whenever you have a variant that is correlated with a particular phenotype, it has to be that variant is impacting the phenotype and not vice versa. Because otherwise you would have to assume that the phenotype changing would actually change the nucleotide sequence of the constitutive genome of an individual, which actually is extremely unlikely to be possible. So once you have DNA variation included into these models, essentially you can start putting the first arrows. So you have DNA variation impacting this metabolite, you have DNA variation impacting the expression, and by doing so, you can then statistically infer potential directional effects or independent effects in the rest of the network. So by integrating DNA variation in a network of phenotypes among a number of individuals, you can imagine these phenotypes can be in the orders of thousands if you integrate quantities of levels of expression of individual genes, levels of proteins, levels of metabolites, many measurements that you can make over an individual. And deriving statistically causal networks, which you can then validate by using either prospective data or using specific experimental assays in specific sets of targeted interaction. Now, at the end of the day, what we're interested in doing, it's kind of a big week, not just my lab, but a very large number of labs, actually many labs are very specific problems in certain areas. In some cases, one is to generally understand what we call underlying biological disease. And the problem with the general thing like underlying biological disease, understanding biological disease is actually very it's one of the easier goals, I would say, in terms of getting to using the genetic information, because in that case you don't have to specifically account for all the variants. You just simply have to say what kind of genes are involved and what kind of pathways are involved in disease. That's a relatively easy goal because that's something that we're already doing with GWAS studies. The other thing that is much more difficult is actually getting an individualized risk profile, which from the genetic data alone, it appears that for most diseases it's not going to be possible, even if I've accounted for the full set of genetic variants, because they don't explain 100% of the variants of the disease. But what you could do is by integrating omics much later in life, let's say a 40-year-old individual, will actually have a transcriptome profile that is a joint effect of their genome as well as their environmental exposures up to that point. And by integrating that, you can actually derive a risk profile that is much more informative than just simply looking at the genotype. And maybe if you do this in a longitudinal way, one can derive models about how the transcriptome profile, for example, in blood or skin or whatever tissue and disease you're looking at, is actually going to be predictive of their outcome because you're looking at much later in life than just birth. Now, my lab has been working on a very, very specific phenotype, which is gene expression. And one of the nice things about gene expression is actually it's present in all cells, and it covers all genes. So we can actually get a much stronger profile about the state of the cell and that particular state. By no means this is the most comprehensive phenotype, and I don't think there is any such phenotype. You know, we can discuss about whether metabolomics or proteomics or transcriptomics is going to give us a picture. My opinion is that everything is required in order to get a complete picture, and everything talks to the other sort of level or layer of information. And one of the things that we've been doing quite a lot is trying to discover what we call expression QTLs. So essentially trying to find genetic variants that impact the expression of nearby genes when we're looking at cis-QTLs, or trans-QTLs where there's a variant somewhere in the genome that through some other interactions is impacting the expression of that gene. And what you get is this correlation of genotype to phenotype, where you, for example, in this case, a GG genotype is associated with lower levels of expression than the AA one. And that we call an expression QTL, an expression quantitative trait locus. Now I'm going to talk about two areas that we're going to use that kind of approach of doing genetics of gene expression in order to address two different questions. One is to actually understand generally how genetic variation, especially from a highly interrogated set of individuals, it's 1,000 genome individuals, is impacting gene expression, and try to get to causal variants and methodologies that one can discover causal variants. And the second thing is I'm going to try to address, to show you some of our recent results in trying to get to regulatory effects in cancer, in colorectal cancer in particular, and try to get to known coding drivers, which has been one of the very difficult areas given the known coding space and how little actually we know about interpreting single variations and somatic mutations. Now the first talk is actually published. There are two papers that are associated with that. One was published in Nature back in September, which is more so the biological insight story. And then there's another important part of the study, which although it's more technical and probably much less biological in terms of insight, it's actually quite important because it describes how we managed to do RNA sequencing in a distributed way in 7 different labs in Europe. And that is actually showing that RNA sequencing technologies and next-gen sequencing technologies are actually mature enough to be distributed and actually borrow throughput from different labs. Now the experiment is this. The project that this was under was a Geovadis consortium. This was led by Xavier Esteville in Barcelona, and this is a European FP7 project that is ending at the end of this year. So what we did, we took 1,000 genome individuals from 5 locations, 4 of them in Europe, from Finland, UK, the CEU sample, which is Northern European from Utah, some kind of complicated ancestry, but they're assigned to be Northern European, and Tuscans from Italy, and also we took a sample from Ibadan, Nigeria, which are called the Yoruba samples. These samples were actually, they're lymphoblastoid cell lines, so these are EBV transformed B cells. And what we did is we grew these cells and we generated mRNA sequencing profiles and small RNA sequencing profiles for these almost 500 individuals, 465 in particular. Almost all of them are actually coming from phase 1,000 genomes, and for those that did not, we use imputation to derive the variants. So we have a very, very dense nucleotide variation map for both SNPs and indels, and a little bit of copy number variation, although the low coverage for copy number variation doesn't give us as much of confidence as one would like relative, for example, to high coverage. And as I said, sequencing was done in 7 European labs, and we had a very interesting replication set where 5 samples were sequenced in all the labs, and about 300 samples that were sequenced in the 6 labs were sequenced and also in our lab in Geneva to assess how replicable the transcriptome profiles are across labs. Now the first thing one can do is to just look at transcriptome QTLs. And transcriptome is not just the expression level, but you can see it in many different ways. You can have a phenotype at the exon level, and correcting for the number of exons per gene, you can ask for how many genes do you have an exon in QTL. And you find that there's close to 8,000 genes that have an exon QTL that integrate some of the exon inclusion variants, which of course have to do with splicing. You can look at whole gene expression, and we have about half of them, so that shows you that just looking at whole gene expression doesn't give you the resolution that you would like relative to exon QTLs. You can look at transcript ratios, which for the moment, that's a very noisy process of deriving transcript quantities from our mRNA-seq data. We have microRNA QTLs, about 10% of the cases. We have repeat QTLs that are nearby the expressed genes. And also, surprisingly, we have RNA editing QTLs. And these are not RNA edited sites that are just DNA RNA differences, but these are bona fide RNA DNA differences that have been known to operate in the sort of the standard mechanism for RNA editing. And about 8 to 10% of them are actually genetically determined. So the degree of editing depends on genetic variation nearby. The next thing we can do is, because we have sequencing data and because we're looking at common variants, is to actually ask what is the enrichment of the lead variant of an eQTL in those different annotations coming mostly from ENCODE and the epigenome roadmap, NIH roadmap. And as you can see, and just looking at the red bar in particular, you can see that pretty much all the annotations are enriched with a few exceptions. And you can actually derive, we developed a methodology by looking at the decay of the signal as you look to the lead variant. So if you take, for example, this case, you see the 15, that's a 10 to the minus 15, that's significant. And you see that there's a red bar, a yellow bar, a green bar, and a blue bar. Now, that yellow bar is actually the enrichment of the second best variant for each eQTL. The blue bar is, the green bar is the 5th ranked variant for each eQTL, and the blue one is the 10th. And from that decay, because you expect that as you go away from the lead variant, your enrichment is likely to drop, and the more likely the first one is the causal variant, the more that drop occurs in the rest of the variants. Ideally, if you had the causal variant in all cases, that drop would be so abrupt that none of the lower ranked variants would have an enrichment. By modeling that decay, we can actually estimate that about 55% of the variants, the lead variants, are actually causal for the eQTL, for the expression effect. And if you look at the African population, we estimate that it's about 74%, because in that case we have linkage disequilibrium that decays faster, so therefore resolution is higher. We can also use allele-specific expression, and that's, I don't know if that's Windows to Mac or Arabic to English or something, but that's certainly not what I wrote. And basically these are two different alleles, and you can take the reads from the two different alleles and basically count the relative abundance of green to red. And you can also do that in terms of structure, so you can say, given that this read maps to that particular position, where's the second read, and you can see that the variability is very different. And you can actually have a 2x2 table looking at significance of this, and I'm sorry you cannot read this, but it's supposed to have something meaningful in there. So basically the idea is that what you can actually derive relative quantities of variance and using a binomial test and corrections for mapping and so on, a very rigorous process and blacklisting of variance for other types of properties, you can derive a set of positions that are differentially expressed. The two alleles are differentially expressed. And Tune Lapalainen, who was the lead author in this project, developed a method that you can discover eQTLs using allele-specific effects. And long story short, we discovered a very large number of those that have a 55% overlap with the eQTLs we discovered with a simple genotypic test. So these are two orthogonal methodologies. One is looking at the cross-population behavior. The other one is looking at the individual level behavior consistent across individuals and among the alleles. And because these are two orthogonal things, you expect that if the truth is there, they would discover similar things, and they do. And then what we wanted to see is that if you take now the variants that are discovered by both, do you actually improve the enrichment in functional variants, in causal variants? And actually you do. So the red bar is the one when you do the genotypic eQTL analysis. If you now condition on those being discovered with an allelic effect, which is again an orthogonal property, you see that the enrichment now becomes even stronger, and we move anywhere from 55%, which was the original estimate, to something like 70 to 75% of estimating that these are causal variants. And that essentially tells you that as you integrate even the same omics technology in a different analytical structure, you can actually get closer to the causal variant, even though you haven't done a specific experiment for that particular causal variant. So this is statistical inference, and of course for any individual variant that is followed up, you need to have a validation. And we can show that in many cases these are actually creating a differential binding. Like for example, for CTCF, you can see that in the null, you get a much lower deviation from the 50-50 allelic binding than you get, for example, for these causal, as expected to be causal variants. Finally, we have effects at the transcript level. So these are what we call loss of function. So you can find, for example, a splice motif that is disrupted, so you reduce the inclusion of the next exon, or you can have a stop codon that is gained, and by that you activate nonsense-mediated decay, and you see that this effect is actually pretty pronounced. So to summarize, I'm not going to go into the detail on that part, but essentially the integration of genome and transcriptome alone could give you a lot more information about the functionality of the genome itself. And what I'm willing to argue is that this debate of whole genome versus exome sequencing is actually done outside of the context of adding transcriptome. And my personal opinion is that if one has actually transcriptome from a tissue that might be relevant, it's actually much more beneficial to do whole genome sequencing, even with a current difference in price, because you can get much more information with respect to functionality of non-coding DNA than you get just by looking at DNA sequence. So I'm going to actually go into now a more sort of translational, in a way, direction, which is to try to integrate some of that knowledge into a real medical problem, which is colorectal cancer. You've heard a lot about it yesterday. So what we did in collaboration with people from Denmark and in the context of the Syscall project, which is another European project led by UC Taipei and Karolinska, is we have 103 pairs of matched tumor normal samples. So these are basically normal from the vicinity of the sample that was taken from the tumor. And this is one of the cases in cancer where you actually know what the normal tissue is like. Because, for example, in breast cancer, you don't know what the normal tissue that you sample is and where the cells that are creating the tumor are coming from. And what we did is we generated RNA sequencing, as well as we took 20 reference tissues to use as sort of a reference for our transcriptome and our other cell type. We generally get a very similar effect in terms of enrichment of over and under expressed, and I'm not going to go into detail about it because this is more sort of type of results that have been shown before, with p53 signaling, cycle regulation, and Wnt signaling as highly pronounced in those differentially expressed genes. We get a typical examples where you have, for example, an exon that is completely missed in the tumor transcriptome, but it's highly used in the normal transcriptome. And when you look at tumor transcriptomes, why the tumor transcripts are highly variable, they tend to cluster together as if they have some very similar properties relative to the normal transcriptomes, even though, as we said, they're genetically matched, which means that the genotypes are shared across these two squares as opposed to within. Now we can again look at allele-specific expression. I told you how we use this. There's a lot of sites that obviously have allele-specific differences that are significant, both in the normal and the tumor samples, and that's not a surprise, but we see many more actually for the tumor because you have an added effect for loss of heterozygosity, which is something that actually generates a differential level of allelic expression. So here we have different categories. You have shared effects, which are in black. You have normal only allele-specific expression. You have tumor-only allele-specific expression, and shared effects that are reversed, which are actually very interesting because what you do is you create another effect in an already existing effect in normal. And as you see, it's much more likely to get an allele-specific site in the tumor than in the normal. Now, one of the things that is interesting about this allele-specific expression is that it is not a property of the population, but it's a property of an individual. So that means that if you have a somatic non-coding variant that landed on the haplotype and actually created a differential level of expression from the original one, that would actually lead to allele-specific expression that is only present in tumor. So what we can do is we can actually summarize these types of effects, and we can look for these allele-specific effects that are only present in the tumor, which they summarize somatic regulatory variants, loss of heterozygosity or copy number amplification in an allele-specific way, which on itself is quite interesting. Now, the first thing we can do, of course, is try to use RNA sequencing to discover somatic mutations alone. So these are protein-coding somatic mutations for the moment. And because the same way you can do exome sequencing, you can do RNA sequencing. And because you're sequencing the coding part, you can discover somatic mutations. And these are the genes that have a significantly increased somatic mutation rate relative to the null. And some of the known genes are actually there. So actually that works. And one can actually say that probably because RNA sequencing tells you what's expressed, maybe that's the thing that you mostly care about. So if exome sequencing becomes quite expensive, maybe RNA sequencing is an interesting proxy, assuming you have enough material and you don't have any problems with RNA degradation. Now you can also go into this tumor-specific allele-specific expression that I was saying before. So you can imagine you have a normal colon where this is a homozygous site. You get a somatic mutation here, and because you have differential, you have heterozygous in the transcript, now because of that somatic mutation and differential binding, you end up having differential expression of this. So essentially, you try to find this mutation, but you cannot, because there's so many of them that you cannot tell the difference between functional and non-functional, but whatever is functional is going to drive differential expression of T to G. So we can use RNA sequencing, that differential expression, as a proxy to regulatory effects. And sure enough, when you correlate the degree of these ASE events, which are now treated as somatic events, these are somatic ASE events, to somatic mutation rate in coding sequences, so this comes from Cosmic, which is a database at the Sanger Institute, you get a very significant and positive correlation. So that means that genes that are already implicated from non-synonymous coding somatic mutations also have an increased level of regulatory effects that are somatic as well. Now you can do a specific test and do all pairs of genes and try to find genes that have an excess of that. And by doing that, you actually get a significant excess of these increased effects of somatic regulatory effects. In some of the genes, you see here a sort of bimodal distribution. So most of the genes don't have anything, and these are only the genes that have no zero count. And many of these genes actually have a very large proportion. So there are some genes that are very different from the majority of the other genes. Now what you can do is you can then take these values, and what I wanted to say first is that this value here, this pi1, is essentially the number of significant pairwise gene tests. So if you take, for example, one individual gene, you test it with all the other genes, you take all the p-values, and you ask how many of them are statistically significant, actually how much enrichment you have in the p-value distribution, and you end up with a score, which in some cases is quite extreme. So that means that if you're 100%, that means that that gene is different from all the other genes in a significant way. Now, if you take that score now, that is on this axis, and distribute it in 3 different categories, other genes involved that are in the pan-cancer, as we say, and genes that are specifically implicated in colorectal cancer, you see that there's an increase in that score. So again, showing that genes that have already been implicated in colorectal cancer or cancer in general have an excess of regulatory effects relative to genes that are just generally not implicated yet or not implicated at all. So we can derive a specific score now in the same way you would do it for coding mutations for what we call allelic dysregulation. So we can look for GADs, genes with allelic dysregulation. And here's a number of genes, there's 71 of them, that actually have an excess of these individual somatic allele-specific effects at an FDR of 5%. And now we can discuss about these types of genes. They actually clearly stand out in that initial score, but they're not representing the full scale. So there's a lot of genes that are different, and probably there's statistical biases for so many genes to be different, but the ones that we actually fished out from the population of genes turn out to be the ones that have the most extreme scores. So generally they fit in that initial context that we were looking for. And they actually have a lot of, these genes appear to have a lot of effects by individuals. So you have some individuals that have up to 23 individual dysregulation effects relative, for example, to some that have only 1 or 2. Now you can go even further and look for something that is even less addressed by the community, which is to find germline variants that were silent in normal, but have become active in the tumor. So essentially what you're looking for is regulatory effects, or EQTLs, that are only present in the tumor sample, but not in the normal. And so here's an example. You have a GA variant here that is not in a regulatory region, because that region is not active. You transition to a tumor state, now that becomes a regulatory region because of some epigenetic change and chromatin modifications. And now, these allelic effect here could actually be detected relative to something that you wouldn't detect before. Now, if you imagine now you have a combination of this effect with a somatic mutation in the coding sequence, you can start talking about epistatic effects between the two. Now we can actually use that, and we discover, sure enough, tumor-specific EQTLs, tumor EQTLs, normal EQTLs, and about 60% of them overlap, which is not surprising. They follow the same patterns that they cluster close to the transcription start site and so on. And they, as I say, they overlap at about 62%. Now, what we can do then is we can ask in the same way we did before for enrichments of the different types of variants, and we can only take the 376 EQTLs that are specific to tumors. So these are only in tumors, very significant, completely flat in normal. So there's absolutely no signal in the normal tissue. And look for the enrichment of that lead EQTL as we did before. And here's the ranking that you see. So this is ranked by the tumor-specific enrichment. You see the red. And this shows the enrichment in the shared one. So the EQTLs that are present in both tumor and normal. And what is interesting is that the ranking that you discover in the tumor is not predictive of the ranking of the shared. So we're actually accessing a different level, a different type of biology by looking at these tumor-specific EQTLs. Now what is even more interesting is if you just look for those that are highly differentially enriched. So let's say, for example, IRX3, which is very highly enriched for the tumor in red, low enrichment for the normal in blue. You can find those, the top ones that you have in this ranking, and ask what are these transcription factors? And these are the transcription factors that you have: IRX3, E2F4, NFL1, 3, TFP2, COX1, and LEF1. And the interesting thing is that all these transcription factors are very significantly overexpressed in the tumor. So the differential enrichment that you generate in terms of the EQTL presence, the EQTL location, is driven by the change in the level of expression, the increase in the level of expression of these transcription factors. So essentially, you transition to a tumor state, you increase the expression of IRX3, and you activate locations in the genome that were previously non-active. And that's why you generate these tumor-specific EQTLs, which could actually be drivers. And we know that they're likely to be drivers because now these genes that have these EQTLs are highly enriched for genes that already have encoding somatic mutation rates and are highly enriched for pan-cancer genes. So what we have is two categories. We have somatic mutations that drive regulatory effects we can generally detect in the sort of the general aspect of looking at allele-specific expression at the individual level, but we also have germline variants which are inactive in the normal, but become active and likely driving the tumor in the tumor state. So this is basically addressing not only the known coding part that has been inaccessible in cancer studies, but is also opening a new category of variants, because so far the community has been talking mostly about two types of variants: predisposition and somatic variants. Now we can have a third category, which are germline variants that are actually somatically active. And that is not actually a very surprising category at the end, but it's an interesting one to be considering both of the regulatory and the coding variant. So with that, I want to acknowledge the big consortium, the Genomic consortium, that actually worked very hard and in a very nice collaborative way to generate the studies that I've mentioned before, and the collaborators in the Syscall project, and of course my laboratory and the funding agency.
I will call upon our next speaker, Professor Sudhir Kumar. He is from the Center of Evolutionary Medicine and Genetics, a Biodesign Institute, from Arizona State University, US. welcome to participate.
Thank you. Thank you. Thank you. So the title of my talk was a little different from what I put in the abstract book. So I will talk about two things, like what Manolis did. And the general title of the talk is now Molecular Evolution in Genomic Medicine, since this session has a genomic medicine theme. And ultimately, in my laboratory, in my research, my primary interest is in understanding the patterns and processes at the level of genomes and genes and mutations that has led to the diversity of life. And that includes, of course, species and humans. And we have diverse phenotypes in humans, a number of them that Manolis discussed. And in general, we see around us the tapestry of life is really dazzling and wonderful. And the institute I am in, Biodesign Institute, our focus is also then, when we learn this information, patterns and process, is to actually harness it somehow to use it for genomic medicine, to making human life better. So we have a dual purpose to our research. One is to do basic science, and the second is to see if we can somehow harness this power of evolutionary knowledge, which is primarily coming from comparative genomics of humans and other species, and for the purpose of medicine and health and sustainability. In my case, the focus is actually on medicine in general. So I like to start with giving you a brief overview of what happens, what kind of projects we do in my group personally, and then focus on one or two topics that are of interest to this audience. So there are three major foci in my group. One is the tree of life, but that includes tree from humans all the way to our ancestors, all the way up to the origin of life. And the second is genomic medicine, as I'll discuss today also. And the third is the developmental complexity, which is what is the phenotypic complexity that we're interested in. Now, unlike I think all other speakers that I've heard over the last day or two here, we don't actually generate any new data. We don't have to go out, get tissue samples, or have animals in the laboratory or any of these things. So we do everything bioinformatically. So my laboratory is basically a bunch of computers, high performance, and a bunch of students. And so what we do is analytics. We do analysis of large amounts of data. So synthesis across many studies, many researcher groups, their output, which is really wonderful given the genomic revolution. And the field and tree of life that we are interested in is the phylogenomics, which I'll talk about just a bit today for a few minutes, which has to do with building the history of life, its organization, its timing of emergence, using genome data. In genomic medicine, the field, what I refer to as phylomedicine, is more about how the tree of life, not just the tree of humans, but tree of life in general, can help diagnose disease and adaptation at the level of humans, individuals, cancers, or anything that we are interested in that has to do with humans. And the third thing is what is called evo-devo, evolution of development, where we are very interested in fruit fly embryogenesis, because that's one of the tractable systems, and image analysis, but I will not talk about that today. And when we do this large-scale analytics, we always have to end up developing new methods, estimate how good other methods are, and so we do a lot of modeling, computer simulations, we do analysis of data. So this is a major area for my group. And when we develop all these methods and models and do data analysis to make discoveries, we end up realizing that other people have similar needs. So we develop a lot of software and databases that are widely used by researchers in the world, including software for comparative genomics, software for SNP ranking, prioritization, diagnosis, and software for actually mining image data using images as a primary object. So today what I would do is I will primarily talk about phylogenomics and phylomedicine and give you just two examples of recently published work in what we are doing. And I'm going to start with phylomedicine. This is a term that I use to basically refer to the interface of genomics, evolution, and medicine. And essentially, phylomedicine is not really about what happened in the past, but rather, given what we know about the past, what can we predict about the novel variants we are observing in our genome? And that's the focus in our case. So in a sense, it is different from evolutionary medicine, a field in which a lot of work is done thinking and looking back and saying, why did we get flu? Or when did this thing emerge only? So in phylomedicine, one of the major focus in my group is the analysis of exomes. And as you know, and Manol has referred to, exomes are essentially a complete collection of exons that actually give rise to our proteins. And of course, we have over 20,000 genes which make proteins. And it's a very minuscule part of our genome, but this is the part of the genome that we know a lot about. And even though it is really minuscule, it's still quite big. There are 15 million amino acids in it. So the number of possible variants that we can have in this exome is very large. And in fact, in this exome, the variants that we are interested in have to do with those that change the amino acid sequence. Therefore, they change the protein sequence. And these are called non-synonymous variants, or NS-SNVs, or you can just in short call them SNPs or SNVs. And this is what I'm going to talk about today. We're interested in many other kinds of variants also, but this is the focus today given the shortness of time. And the reason why we are really interested in that is in the last few years, exome sequences has really become very routine and much more accurate than it used to be. So if you look at the trend of discovery of exome SNPs, especially the non-synonymous kind, which make protein change, it really has exploded just exponentially. This you can compare to the SNPs that are found in the whole genome, which is also expanding very quickly in absolute number, but it's still a bit more linear than exomes. And it's because exome sequencing is, as I said, routinely done in cancer groups, routinely done for patients for various reasons. And so this is the focus we have, and it's an important focus and an important part of our understanding. So the grand challenge in genomic medicine, and there are many, many of them, and one of them is, if you were to think about individual point mutations being important for disease or make a significant contribution to disease, then how do we identify them? In any individual, you're walking around with as many as 7,000 to 8,000 such variants. Some of them are de novo in the last generation or two. Many of them are family, some are population specific. How are they going to impact, or how are they impacting your health, your ability to metabolize drugs, and all of those things? That's the grand challenge. And so I got interested in this work more than 10 years ago, just doing very simple evolutionary biology, asking the question, what is the human genome variation which is involved in disease, especially at that time, primarily Mendelian diseases, have to do with long-term history of humans and chimpanzees and gorilla, orangutan, all of these species, which is among species. So we decided to just do simple evolutionary biology, try to understand it. And so over the 10-year period from 2000 to 2010, we just tried to develop lots of knowledge about it and made some fundamental discoveries which were, in retrospect, quite obvious. But nonetheless, they were discovered at the time. But during this time, in the last 10 years, there were another group of researchers, unlike us, who were actually developing methods for diagnosing which particular exome variant will have disease potential, or which SNP will make potentially a disease contribution. And really wonderful software like Polyphen and SIFT were produced, which are now used 1,000 times or more than that every year. Today, everybody runs all their variants through them, because you find so many variants and you have to pick a few. And in the last couple of years ago, we began thinking about these kinds of variants and these kind of methods. And we saw there was a new paper that came out in 2011 in American Journal of Human Genetics, which is a Condell software, which took a bunch of these methods together and combined them in a wonderful package, made what they call Condell. It's a hybrid program. And then based on the best features of all these methods, they were able to presumably increase the accuracy. So if you look at the topmost curve, which is a curve which flattens out, goes very quickly up and flattens out, it shows that they could really do an amazing job. They could do true positive, correctly identify disease variants, potentially, 90% of the time, with only 10% false positive. This is an amazing performance. And so it sounded very interesting to us to explore if this is really true. And so we had some understanding of evolutionary patterns. Many of these people actually use sequence alignments. So we expected the things to work out really well for them. So the question I asked very simply was, we asked, if you look at the positions that are most important functionally, that means they have barely changed ever in the history of vertebrates, 500 million years. So evolutionarily, no changes have occurred at these positions at amino acid level. And experimentally, if you look at it, most experimentalists actually look at positions that are unchanged among species because they think this is very important. These are important sites. So we asked the question, what is the accuracy of these methods for these positions? You know, it's a very simple question. And given that the fact that it should be 90% accuracy based on what we saw in the literature, we did the analysis. Unfortunately, the results were not really that good. It showed that all of these methods, like polyphen 2, Condell, SIFT, they suffer from severe problems. They incorrectly identify non-disease causing mutations to be disease causing at an astonishing rate of 70 to 90%. So to us, this is quite surprising, and we tried to sort out why this happens, and we did sort it out, and I won't go into detail a lot about that. But what we realized is, okay, so there is such a huge error. This actually means that anybody who used these tools in the last 10 years and picked the most dangerous SNPs probably is wrong more than half the time, or maybe 90% of the time. So this is one of the reasons why we have to really know this kind of problem. So this sort of an evolutionary approach to diagnosing error has not been taken before. And so what we thought is we will develop a method that sort of fixes this problem, because ultimately that is our interest, is developing better methods. And so what we developed is a new method published late last year called EvoD, in which we use fundamental evolutionary properties of the position where the mutations happen or variation is found, and the actual variation in a propensity, biochemical as well as substitutionability. And basically is using 12 primary parameters, predictive parameters. We did what we call, what you might all know about is machine learning and predictive modeling using this data. And we use training data like disease, known disease data, and not non-disease, neutral variants presumably. And we were able to build a mathematical model, a simple one, really regression model ultimately, and it produces a single impact score. It's a number. If a number is 100, it's a very, very bad mutation, like it might cause a disease. If a number is 0, it is really benign, what we call neutral variant. And so if you actually take the 1,000 genomes data that Manol has discussed and put it on x-axis as an impact score and y-axis as the frequency of alleles in the 1,000 genomes data, what you find is a nice result, that there is a negative relationship, and that actually we found that a score of around 50 or so is a good cutoff between, you know, truly benign and potentially problematic variants. And so when we use this method, we were able to actually improve upon the error on polyphen and Condell and SIFT, all of those methods. So in fact, the black bars show, compared to Condell, if you compare to Condell, we have reduced its false positive rate from 90% by 70%. That means just false positive rate is 20% approximately. If you do compare to polyphen, we reduced its false positive rate by 50%. So essentially, around 20% error cannot be removed. But other than that, this method actually takes care of most of the bias that exists in current tools. And so, and this is true for all kinds of variants, high or low frequency variants for the human data that we know about. And I should tell you that we used the same exact data set that was used by polyphen and Condell for training their models. They're absolutely the same information. This way the results and comparisons are absolutely the perfect comparisons with no changes in data sets. And then actually we applied this to complete exome data for many individuals. We took actually 8 individuals from a cell and egg paper, and we applied this. And if you actually look at positions that are extremely well conserved, which is the left bar, middle one is well conserved, means some change is allowed by evolution over time, means slight flexibility exists. And last one is a lot of changes are allowed. And if you look at all the alleles found in heterozygous genotype, you find that around 60% of the variants are neutral or non-affected, and around, I would say, 25 or 30% show problematic signs, which is by red coloring. But heterozygote, it seems like, have, across these 3 categories, not much difference when it comes to percent variants. Now, there are more variants found in less conserved sites and ultra-conserved sites, but if you look at fractions, they're pretty similar, and it kind of makes sense because we know lots of variants of functional impact are recessive, and they just sit around waiting to be expressed when they become homozygous. So what happens when they become homozygous is, you see that first of all, homozygous have a number much smaller of bad than heterozygous. And the second is the homozygous in fact show a difference between 3 categories: ultra, well, and less conserved. In less conserved category, there are more homozygous have more disruptive looking variants than ultra conserved. In fact, in one exome, in the homozygous, the deep red you see is just one variant. So at a very high probability of significance, there's only 1 or 2 or a few variants in an exome that are really that bad at ultra-conserved sites. So this was actually made sense from the purpose of the way alleles act, dominance of wild type versus recessive alleles versus mutants, I should say, and their recessive nature. So it was a very nice result, reconfirming what we expected. So just to complete on that, if you're interested in using a tool like EvoD, we have recently developed a software, which is what we always do, where you can in fact take your data from any exome survey, thousands of variants, one variant, you can enter that data through this tool, and we have programmed this within a software that we have, which many people use, called Mega, and it's available as a standalone software also, also as a web service. And it also provides you with polyphen 2 and SIFT predictions. So this way you can see all the predictions, which is not really about just our method, but it's about you having overall good result. In fact, consensus results tend to be very highly accurate across 3 methods. And also we are trying to have researchers in population genetics who are using this variation data also look at this data from the evolutionary perspective, long-term history. So now we integrate long-term history with short-term history in our display, Tree Explorer, where you can examine exactly where that variant occurred, if it ever did, in the history of vertebrates. So this is a software tool that you might find useful for your research. And I should also close this part of the presentation by telling you that we are also looking at nonsense variants. We have a new method for that, which is more accurate than any other method published to date. Insertion deletions, with and without frame shifting, and also even synonymous variants, because we now know a number of synonymous variants are involved. But I will talk about that maybe in the future in any similar venue. So that was the first part of my talk. Hopefully I didn't take too long. The second part is phylogenomics, and this is the part that has to do with the tree of life part. So in genomic medicine, we are constantly estimating when did certain infections occur, when did a HIV strain arise, and all of this requires timing the origin of a strain based on the contemporary sequence. So we have been doing research in this area for a while, and our primary focus in the past has been on species evolution, but the same method can be applied to strain evolution. Ultimately, they are sequences, especially if you're thinking about viruses and bacteria, which have a different sort of a history. So the field of phylogenetics has to do with really trees, using phylogenies and converting them to time trees, where there is a time on the scale. And there are many methods available, multi-diff time, MCMC tree, BEAST. As you get cancer mutations and all of these data, you can actually have large number of sequences, and you want to find out when these mutations happened and what was the time. So you have to use these methods, and people are using them. The problem, as first I should mention here, is a lot of people are very interested in times of divergence. In fact, if you look at over time, cumulative number of studies published, is just increasing every year by a few hundred. So that's what people are publishing. And the number of species for which we know times is almost as many as 50,000. So you can sort of build a tree of life scale to time based on these diverse species. So if you're interested in knowing about any pair of species history, human-mouse divergence time, what do people say? What do the scientists who are working in that field say? And you're a molecular biologist. You can actually go to our website called timetree.org. We even have an iPhone app, an iPad app. You just put in human-mouse. It will tell you every study published and what time have they found. And this way you can have a quick look at the field without having to know their nomenclature and their complexities. This will basically give you the information. So it is a lot better than Google, where you have to read the paper. Nowadays, no one has time to read the papers, as you know. Everybody's very busy. So this is a resource that you can have a look at. But anyway, the challenge in the computation is in timing of strains or species is it's computationally very demanding. For example, for 400 sequences if you have, the program that many people want to use, BEAST, will take 92,000 days. That's a long time. You need supercomputing square or something for that one. If you had 1,000 taxa or sequences, it will take forever. You just will never finish. So many people just subsample the data or try to deal with it. So the other problem is many tools require knowledge of the rate of how the rate is changing over time in a tree because of the adaptation, because of the strains, because of random nature, and then you have to know when did certain nodes in the tree have a common ancestor to scale everything to time. So what we did is we said, well, what if we don't have any of that stuff? Can we really come up with a method that was fast and easy to use and doesn't require any of this information? And so we did. We published that late last year also. It doesn't require any of the stuff that everybody else requires, which means it is not based on those information. It's more algorithmic than a statistical method, and it's really fast. MC2T, which is the fastest method out there in Bayesian world, ours is a likelihood method. It will take 10 days for 1,000 sequences. Ours will take 3 hours. So it makes it really possible to calculate all of these things very quickly. And this is the published stuff. I won't go into how it is, but essentially our method produces as good estimate as the true times if we have large enough data set. And in fact, for the same data set, you can compare our method to MC2T or BEAST, and we in fact do better because our curves are much more centrally located than MC2T, for example. And if you have a directional change of rate in one lineage, which could happen because of adaptation, for example, our method is still doing well for both inside and outside of that clade, and unlike other methods that are possible to compute, but they have a bias in their methodology. So basically, Bayesian methods, the way they are constructed currently, are slow and actually inaccurate, mostly extremely biased. And actually, we very recently, just in the last few months, extended this method where we not only use species times, but also if we know the time of sampling of the sequence. So HIV sequence is sampled from the same patient or multiple patients over time in the last 10 years. That's shown by the x-axis. And then, so therefore, it's almost like you are sequencing ancient DNA. And you want to know when did they diverge from a common ancestor. So in this case, the problem becomes slightly different, but we were able to redo the REL-time methodology. And in fact, if you look at the REL-time that we produce within a minute compared to BEAST, which takes many days, and the time estimates are practically identical. So it's a very fast solution to produce divergence time estimate for any type of data. And in the case of cancer, that would be very interesting in the future as you have sequences from different strains, from different parts of the different tumors in the same individual. You want to know when did this actually get here? When did these cells leave the parent cluster in time? These are the kind of method that people will have to use to estimate that time. And again, we have recently added this to REL-time to MEGA-6, which is the paper just published in MBE. So you can easily utilize this method in the current version of MEGA, which is online as a beta version. will be very soon complete. So this gives you an idea and the error bars on estimating the time of the origin of strains or species. Our method works for both equally well. And so I want to finish by saying that, I use the same exact principles of molecular evolution for all the problems. I have one toolkit I learned, and actually Manolis and I, we both went to graduate school at Penn State at the same time. And what we learned is population genetics and molecular evolution. He does more pop gen and I do more molecular evolution, but ultimately they are two facets of the same problem and the same pattern. And so what we are trying to do here in my group is to work on the field of phylo-medicine, where I insist that long-term history beyond humans is very informative about human variants. It's really an important thing. And evolution should be seen as a technology, not just as a process or anything for solving problems today. So I think of evolution more as a technology than as a field in many cases. And phylogenomics, of course, has solved many of our problems and is going to continue to help us build the history of strains and species and their timing. And ultimately, we are translating this knowledge into practice for practical tools that I think used widely now by students and researchers everywhere, and even medical fields. So I want to finish by thanking a number of my associates and funding agencies for their support. Thank you.
Thank you very much, Professor Sudhir, and for a very interesting perspective. Of course, it's unique and has led us to a very important question. The evolution of human didn't start from apes or not. But anyway, we will leave that afterwards. Again, as I said, that we will leave, if time allows,